Using Clino fragment for DNA sequencing, that is enzymatic sequencing, we are going to see the steps involved in the complete process. Step number 1, अगर हम broadly classify करें और categorize करें, यहाँ पे हमें 3 different किस्म के reactions run करने हैं. The first one would be the annealing reaction. The second one would be the preparation of the labeled tubes, the 4 tubes to perform the 4 labeled reactions, and the third one will be the extension or termination reaction. So the first step is to prepare the template along with the primer for annealing. For that purpose, we are going to add single-stranded DNA in the volume of 5 microliter along with the oligonucleotide primer 4 microliter, and the TM buffer is used here, that is 1 microliter. So the total volume will make up to 10 microliter. This mixture, after closing the tube tightly, this mixture is incubated at 55 degrees centigrade for 5 to 10 minutes to complete the annealing process. During this 5 to 10 minutes incubation period, we prepare the 4 tubes which are labeled as A, G, C and T, which are known as labeling tube, or to proceed further for the sequencing. For that purpose, we are going to add 4 microliter of appropriate dideoxynucleotide triphosphate in each tube separately. For example, for a tube which is labeled A, will get 4 microliter of DDATP. The tube which is labeled C will get 4 microliter of DDCTP, and so on. From step number 2, after incubation of the annealing mixture, annealing is completed. Now we are going to add DATP, radio labeled DATP, 1 microliter in it, Klinov enzyme 2.5 units in volume 1 microliter, and DATP 0.1 millimolar. This is normal DATP, this is not the labeled one, but it is going to be used only if we used radio labeled DATP and its phosphate variant. अगर हमने sulfur variant use किया है, alpha 35 sulfur as a radio label use किया है, तो उस सूरत में हम water add करेंगे, DATP add नहीं करेंगे. Let me make it clear here that if this is our component, this one at this stage or this one, then we are going to add this nucleotide in the given volume. और अगर हमारे पास जो component है वो हमने use किया है alpha 35 sulfur, then we are going to follow this step. That is, we are going to add water in it. The enzyme है Klinov enzyme, because this is an enzyme, it should be properly stored at given temperature. That means it should be stored on ice all the time, and if it is not being used, it should be stored at -20, क्योंकि long term storage ice के ऊपर जो है, that can lead towards the loss of activity of the enzyme. Then in the next step, we are going to add 3 microliter of the mixture from the previous step to each of our label tube, that is C, T, A and G tube. But at the same time, we will try that our mixture should not go to the bottom. अगर आप इस drawing को देखिए, that this is the tube in which we have our reaction mixture. We are going to drop the step number 4 mixture, the 3 microliter, on the wall of the tube. Because this contains the radioactive material, it should not go and directly touch the other liquid material or the mixture. For mixing, we are going to perform the centrifugation. Few seconds centrifugation at 2000 RPM is going to mix the components and is going to initiate the extension or termination reaction. We are going to incubate all these 4 tubes for 10 to 12 minutes at 37 degrees centigrade. Once 9 to 11 minutes of this incubation are passed, 9 to 11 minutes of this incubation are passed, then we will transfer 1 microliter of chase mixture to the side of each tube, that is A, T, C and G. अगेन यहाँ पे ये बताना ज़रूरी है कि ये चेज मिक्सचर जो है इसको आपने 1 माइक्रोलीटर को ट्यूब की वॉल के ऊपर डिपॉजिट करना है. And as soon as 12 minutes incubation gets completed, a brief centrifugation is going to be done so that chase mixture goes to the bottom and gets mixed up with the components already present there. Now after this brief centrifugation, we are going to go for another incubation for 10 to 12 minutes. That 10 to 12 minutes incubation is basically for the action of the chase to be performed. And again, after 9 to 11 minutes, we are going to add 6 microliter of formamide loading buffer on the wall of the plastic tubes, on the wall of the microfuge tubes. Again, we have to wait for 1 minute so that 12 minutes of the incubation are completed. Then we subject it to the centrifugation, a brief centrifugation, so that the formamide loading buffer can perform its function and can go towards the stoppage reaction. Towards the end of the incubation period and centrifugation that stops the sequencing reaction, we have 2 choices. Either we proceed further to store the sequencing mixture now at minus 20 degrees centigrade for 5 days, or we proceed further for denaturing gel electrophoresis so as to perform the sequencing of the DNA. And for that purpose, we are going to heat denature 2 minutes at 100 degrees centigrade and then quick cooling on the ice. And out of the tube, we are going to take 3 microliter to run in 4 different wells of the gel, and we read the sequence from the sequence gel based on the molecular weight from bottom of the gel towards top of the gel.